Okay, let's get into chapter 7, where we're going to talk about microbial growth. Remember that this isn't meant to replace everything that is in the longer lecture. This is just a rundown of the major concepts. Anything's fair game in the lecture slides, just saying. So microbial growth, here we are talking about basically whatever factors might favor growth of certain bacteria or what they might need in order to grow and everything like that. To begin with, the major elements associated with microbial growth are going to be, I have it abbreviated as CHOMPs, but CHONPS. So carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. Those are the big 6. Definitely be aware of them and be able to associate them with the fact that organisms need these things in order to even just friggin' exist. Okay. We will talk more about, I guess, the life cycles, the element cycles a little bit later on in like 24 and 25, chapters 24 and 25, but be aware of those. Okay. So how are we going to be getting these nutrients? Well, heterotrophs like you and I, we source our carbon from organic molecules that typically come from other organisms that fixed that organic molecule to make it an organic molecule. Because autotrophs, like plants, they use inorganic carbon. In this case, we're talking about plants, carbon dioxide as their carbon source. They don't get like their carbon source necessarily from their organisms or something, or other organic compounds. They fix that inorganic carbon and create organic molecules from that. And that's where we get our carbon from, is by eating things like plants that have already done that for us. Phototrophs are organisms that get their energy from light. So here you can already see that we're talking about the difference here, where our carbon comes from. So carbon source, and then where our energy comes from. Carbon sources, energy sources, different things. Okay. Phototrophs get energy from sunlight. They're able to basically harness that into the next few steps after that to create energy forming molecules. And then chemotrophs get their energy from chemicals. So we essentially break down like what we do, which is breaking down glucose and using that breakdown of those chemicals to essentially create. Sorry, my stomach is really loud. I'm sure you can hear it growling. I'm not going to go take care of that. I'm not going to go eat something right now. I want to get this over with. So, anyways, breaking down some chemicals to harness the energy that we get from that breakdown into another kind of chemical we can use for other things in the cell. That's the idea with the chemotroph. Okay, so that's heterotrophs, autotrophs, phototrophs, chemotrophs. So a photoautotroph is something that uses energy from light and the carbon is inorganic, CO2. So that's like plants, right? So you can put these words together and ascribe to them the appropriate meanings, such as what we would be, which would be a chemoheterotroph. So we get our energy from chemicals, like our food, and we get our carbon from other organisms, like our food. Right, a growth factor is something that must be provided to organisms for them to be able to survive. So it cannot be manufactured by the organism itself. We have talked about this previously with scurvy when we talked about vitamin C being necessary for humans. We can't make it in our bodies ourselves, so we have to have it provided to us in our diet. Saprobes are organisms that will digest dead things. Dead stuff is called detritus. So anything that's dead and decaying, that's called detritus. Or detritus, depends on however you want to. I don't care how you pronounce it as long as you can identify it on a test. Okay. Halophiles, these guys are going to flourish in salt. Halo, this is literally referring to like salt. It's like a halogen, essentially. And then we have thermophiles, temperature loving, right? So thermophiles grow well at high temperatures. We also have organisms that do well at lower temperatures, like the psychrophiles and the psychrotrophs, but those are at lower temperatures. All right, so this chapter is where we get into the concept of diffusion, because we start to understand a little bit what the usefulness is of like desiccation and things like that for controlling microbes. Diffusion is a term that refers to the movement of molecules from an area of high concentration to low concentration. Osmosis is just the diffusion of water molecules. So if you have a membrane that water can cross over, but other molecules can't, then that would be something called osmosis. So let's say you have water can go in and out of here. So this is important because we're going to talk a little bit about other things that cannot go across. Color. Okay. Okay, so a solute, these are molecules that are dissolved in the solvent. So that usually in our case in biology, we're talking about water being the solvent because that's the solvent of life. Then this membrane that I've drawn here is a membrane with selective permeability. So everything, not everything can go in. Where is it? There it is. So let's say these guys. These guys can't cross. Whatever this is. I don't know what this is. What do you want to call this? Sugar. Sugar can't cross, right? Water can. So that is selective permeability. Seems simple enough, right? Okay, so isotonic, hypotonic, hypertonic. So let's say that this is inside and this is outside of a cell. So we've got a ton of sugar on the outside of the cell and none on the inside. Ideally, in a perfect world, you would want to move these sugar molecules inside to equal out what the concentration would be, but we can't do that. We can't do that because we are selectively permeable. Only water can move, okay? So essentially what the body or what the cell will do is, let's just say there's a little bit on this side. It doesn't matter really, but it's just a whole lot out here, okay? What the body will do is try to push water out to make the concentration of that the same as the concentration outside. Okay, so that's what it wants to do, because outside in this case, in this picture, outside is hypertonic. Compared to the inside, okay? So the solute, the green, is higher on the outside. This creates osmotic pressure, which means that, you know, essentially it's drawing the water out of the cell here. So the water will move out and the cell will shrink down as a result. If this were reversed, if this were outside and this one were inside, then basically this would be called hypotonic because there would be less. That makes sense. I think that makes sense. So I do a little bit more detailed job of explaining this in the lecture, FYI. So I would go back and review that if this doesn't make sense to you for some reason, or if you want me to explain this further in the actual review. When we do the review before the exam, and let me know, put this on your list. Okay, because I'm going to move on. All right, facilitated. So we know we have selective permeability and the water can move back and forth in these membranes that we have surrounding these cells. Well, what if we need to move the green blobs in and out? What if we, want to take care of that? Well, if we are moving the green blobs from outside to inside, as would be depicted, from outside to inside, the green blobs, see how they're higher outside, we want to move them inside. That's going to be movement by diffusion, right? From high concentration into low concentration, but they can't cross the membrane because it is selectively permeable. So we can have these molecules that we can insert like a channel that the things can go through there. They'll still move by diffusion. It's just now they have what we call facilitated diffusion. That is a spot where they are being helped out to be able to cross that membrane via diffusion, but it's still diffusion. What if you need to get rid of this guy, though? What if we don't want him there at all? Well, then we have to go against the gradient, right? So if you're going to an area of low concentration to high concentration and trying to force more out, that is going against diffusion. That's going against the gradient. So then we need something called active transport. And we're going to need to use energy in order to force something against that gradient. We can do that. We can pair it with using ATP or something else associated with energy and force that to go on the other side. And that would be active transport, moving against the gradient, but it requires energy. Okay, so another way we can divide up microbes and how essentially we have defined them based on their living conditions and what they need to live and everything like that. We know we need oxygen to survive, right? To deal with oxygen products and in microbes, so some of these guys are pretty good at surviving without oxygen. We have the ones that have to have oxygen like we do. Those are called strict aerobes. They have to have oxygen. Tuberculosis is an example. Then we have facultative anaerobes. These guys live fine with oxygen. They do well with oxygen. They make more energy with oxygen. But if you deprive them of oxygen, then they will go through something called fermentation, which we'll talk about later. And we have microaerophiles, which can survive with very, very little oxygen. We have aerotolerant anaerobes. These guys don't need oxygen at all. Survive without oxygen, but can survive in the presence of it. And then obligate anaerobes. These are guys that have to live without oxygen. They don't use oxygen at all. And oxygen, in fact, usually will kill them. Oxygen, we don't think about it a lot, but oxygen actually has a lot of like toxic byproducts associated with it, whether that is like superoxide ions or ozone or anything like that. These are oxygen molecules that can cause damage to the cells. Organisms that are adapted to using oxygen have the ability to deal with that. Anaerobes, the ones that live without oxygen, some of those guys, the obligate anaerobes, they have no ability to defend themselves against toxic oxygen byproducts. So that is something to think about as well, as far as like treating those guys, or even just testing them in a laboratory to find out if they possess the certain enzymes that are protective against an oxygen byproduct. So, all right. Next, we have how organisms like interact with one another essentially in their lives. We have symbiosis. This is whenever two organisms' livelihoods are intertwined somehow, the good or bad. So we can break that down even further to commensalism. This is whenever one organism benefits and the other one is neither harmed nor helped. Then mutualism, both organisms are being benefiting from the experience. Both organisms are getting something good from the experience. A lot of our gut bacteria are these guys. And we have parasitism, where one organism is stealing resources from the host and causing harm to the host, and then the host is being harmed. So that's parasitism. And then we can look at the different, as far as looking at microbial growth and the phases of their growth. When you first have introduction of bacteria into a medium or into a host or whatever it is, they first have to kind of adjust to their environment. So we see this introduction. So this is the point of introduction, and it's flat at first. This is what we call the lag phase, where they're just basically gathering ingredients and getting used to their new environment and getting ready for division and starting up with replication of DNA and everything like that. Then we have the exponential phase, or the log phase, where we have exponential growth and division, which speaks for itself. Then we have the stationary phase. This is essentially where growth and death will equal out with one another, as well as, we're running lower on nutrients and we aren't growing as much and stuff like that. And then we have the death phase. We've run out of nutrients and the organisms die. They don't live forever. So that's basically that. So that is it for the microbial growth, introduction of that stuff. The next chapter is my favorite. It is the Metabolism chapter, and I cannot wait to tell you about it. And I'm sure you heard about how excited I was in the video. I don't know if I was excited in the video, but I'm excited now. So hopefully you guys got something good out of that, and then you can come to the Metabolism lecture next and get to hear about that. I cannot wait. It is so fun. So I will talk to you guys. I'll talk to you guys in this chapter, and I'll see you then. Okay.